Hello, I'm Suhail Alam. I'm a biology NEET faculty, and I mostly teach class 11 and 12 both that biology portion. Okay, so today we'll talk about the biotechnology application in our agriculture. Okay, so we can use the biotechnology in different way in our agriculture, but today in our class we talk about the BT cotton only. Okay, so today we'll talk about only the BT cotton. Okay. So what is BT cotton actually? Okay, it's a plant. What kind of plant it is? It is a transgenic plant. Okay, it is a transgenic plant. What is mean by transgenic plant? So plant whose genetic makeup is we can modify by the genetic engineering. Those kind of plants are called what? Transgenic plant. Okay, so how we can modify their genetic makeup? Okay, so they have own their genetic makeup. Okay, but here we can take a desirable gene. Okay, we can take a desirable gene, and this gene one we can introduce with a vector. Okay, with a vector we can introduce, and this all together we can introduce in a host body. Okay, in host means here we will be insert this all desirable gene with the vector in our cotton plant. Okay, so that's why here a cotton plant have their own genetic makeup. Okay, but we inserting another gene that we need. Okay, so this all together we just modify their genes. That's why they're known as what? Transgenic plant. Okay, but why should we do this one? Okay, what the need of that? So why should we do this one, and how we'll do this one? Okay, so this all together question we'll get in our previous discussions. Okay, so now we'll find out the answer of that to question. Okay, so here why and how will we do this one? So now let me discuss here. So here is a bacteria. Okay, there is a bacteria who is known as what? The Bacillus thuringiensis. Okay, thuringiensis. Okay, so this Bacillus thuringiensis is a bacteria. Okay, is a bacteria who have their own genetic materials. Okay, they have their own genetic material, but in their genes, okay, in their all total DNA material, they have a particular genes. Okay, and this gene known as what? The cry gene. Okay, this gene is known as what? The cry gene. Okay, and this cry gene are responsible to make some kind of protein. Okay, so this goes in the transcription translation. After they are make some kind of crystalline protein actually. Okay, they make some kind of crystalline protein. Okay, they make some kind of crystalline protein. Okay, and this protein also known as what? The insecticidal protein also means this protein have the ability to kill the insect. Okay, they can also kill the worm, they can kill that beetle also, they can kill that flies also, they can kill our mosquitoes also. Okay, lots of also caterpillar also can be die if this kind of toxin goes in their body. Okay, so this toxin is what actually? This actually not toxin, actually this is a protein. Okay, but when they are active, okay, when this protein will be active, okay, this protein when will be active, they become toxic. Okay, so this will be toxic. Okay, they will be toxic, and this toxic known as what? The BT toxin actually. Okay, they become an BT toxin. Okay, BT toxin actually. Why is BT toxin? Because this toxin is made from where? Bacillus thuringiensis. Bacillus thuringiensis short form is what? BT. Okay, so here a toxin that are formed in the BT's body. Okay, that's why this is known as what? The BT toxin. Okay, so how they actually working? So when suppose any kind of nematode or anything, okay, any insect, suppose this insect one just take that toxin in their body. Suppose this toxin by chance, okay, this toxin by chance goes inside of their body. What will be happen? So this toxin will be attached with their gut wall. Okay, and they will be make the swelling of that, and also they make a hole in their gut. Okay, so as a result this insect will be altogether die. Okay, no, they will be not able to survive. Okay, so this thing can be happen. Now the question is came ki BT is a Bacillus thuringiensis. Okay, the bacteria, so they making some kind of toxin, and toxin is not good for everyone. Okay, so here toxin they making, so this is not harming in their body. So the answer is what? Ki here this bacteria's body pH is so low. Okay, they are acidic, so that's why in their body this toxic one, that protein one actually, the crystalline protein one, not able to be active. Okay, so that's why they're some mostly behave as what? Protoxin. Okay, that's why they're not harming them. But when this protein one goes inside of their body, and in their body the insect body have the higher pH, the alkaline pH are there, so when they goes inside of their body, they are just pH is high, and this time that protein will be active and they'll be destroy their body. Okay, so like this way this BT toxin is working. Okay, now how we can use this one? Okay, so here normally suppose a cotton plants is there. Okay, they have their own genetic makeup. We don't do any kind of genetic makeup or something, okay? So we don't do any kind of things, okay? So it's a normal cotton plant seed there, okay? So they have their leaves, okay? Suppose here suddenly a caterpillar came, okay? Caterpillar came, so they'll be do what? They'll be take their they mostly feed on that the leaf of the cotton plant, okay? So mostly they all together leaves will be eaten by that caterpillar, so they obviously harming that cotton plant, okay? So now how we can protect them, protect that cotton plant from that caterpillars, okay? So now what we can do? So in that plant now we can introduce that our desirable that gene, okay, the cry gene, okay? So what we'll do here? So to introducing the cry gene, first you have to isolate our gene, okay, the cry gene I'm isolating, okay, and this one we have to insert in the plant body. So here we need a particular specific plasmid. This is mostly useful in the plant. This is what? The TI plasmid. So now this target gene, okay, we have to introduce it what? With our TI plasmid, okay? Now all together our plasmid and also that our desirable gene we have to introduce inside of our plant body, okay? So when they will become inside of that plant body, the cell they will become in the cells, okay? Now cell will be divided and every cell of that plant body will be get that gene, okay, cry gene, okay? So now they also able to make the same toxin that are able to make that our bacillus thuringiensis, okay? So now after that when a insect will become here to feed on them, okay? Suppose the insect is came, okay? Suppose the insect is came, okay, to feed on them, okay? So whenever suppose they're just taking that feed on that leaf, okay, and this leaf have that cry gene also, okay, they are all total make that toxin one, okay? So now this toxin when they'll be feed here and this will be goes in their gut also, okay, and the same thing will be happen, they'll be destroy their gut and all total this insect will be die, okay? So like this way without using any kind of insecticide, any kind of pesticide, we are able to control that caterpillar one, okay? So this is the actually application of our biotechnology, okay, where we can without using any kind of the pesticide insecticide we can able to control our cotton plants, okay? So now hope you're getting that idea about why should we use that our why should we use that kind of biotechnology in our agriculture, okay? So avoid the uses of insecticide and pesticide. So hope you are getting some idea from that class. Thank you.